You are muted, Si Mohammed.
Greetings to you all. Happy to have you here among us for this 8th webinar, which is contributed by our distinguished professor, Mustapha EL BOUHSSINI. Dr. Mustapha EL BOUHSSINI joined UM6P, Mohammed VI Polytechnic University on the 1st of February 2021 as a professor of entomology and program lead of the Biodiversity and Plant Science program. Dr. EL BOUHSSINI earned his PhD in entomology from Kansas State University in 1992. He started his research career as entomologist at the Dryland Research Center in Morocco, located at Stade, before joining the International Center for Agricultural Research in the Dry Areas at Aleppo, Syria in 1996. Dr. EL BOUHSSINI has also been an adjunct faculty at Kansas State University, the entomology department, since 2005. Dr. EL BOUHSSINI has made exceptional contributions to the development of integrated pest management options that are increasingly used and scaled out, particularly in the West Asia, Central Asia, and North Africa, generally termed by our friends of ICARDA as Siwana region. His major research focus has been on IPM of key pests of cereals, food legumes, date palm, and cactus. Dr. EL BOUHSSINI has been playing a significant role in human resource and capacity development, including through giving training courses, mentoring individual trainees, and supervising the research of more than 20 master's students and 16 PhD students. And further than that, over 500 junior scientists and technicians from Siwana region have benefited from his training courses. Dr. EL BOUHSSINI has been the recipient of several awards that have recognized his achievements in the scientific field of entomology. Totally, he has obtained 15 awards, an impressive number of awards, between national and international awards. I would like just to cite the international ones, and I will start by the first award that he obtained in 1998 at ICARDA, where he was honored as the Scientist of the Year. The second international award was granted by the Plant Protection, the International Plant Protection in 2007 as a distinguished, an award of distinction from the International Association of the Plant Protection Sciences. In the year 2014, he has obtained the Distinguished Alumnus Award from the Kansas State University Department of Entomology. And in the same year, 2014, he has obtained the Distinguished Scientist Award from the International Branch of the Entomological Society of America. And lately, in 2018, he has obtained the award of Lifetime Achievement Award in Plant Resistance to Insects from the International Association of Plant Resistance to Insects. So today, Dr. EL BOUHSSINI will speak to us about a very interesting topic termed the progress in host plant resistance to insect pests in North Africa, West and Central Asia. and he will go through some crops as example of his studies and research outputs. Dr. El Bouhsini, the floor is yours.
Thank you so much, Professor Boulif. It's really a great pleasure to be with you here today and talk to you about an area of research that it is really in the heart, in my heart. And this is what I've done, I would say, the most in my career. And so I'll just give you a flavor of what I've been doing all these years, working on insect. Of course, now my area of specialty has been enlarged and become now the integrated pest management. But I would say more than 70% of my time has been on host plant resistance to insect. So I will cover a few examples to show you what I have done in collaboration, of course, with partners from the region, advanced research institutions, etc. Now, this is where I was. This is North Africa, West Asia, and Central Asia, Aleppo. ICARDA used to be here, the International Center for Agricultural Research in the dry areas used to be here. But now, of course, it has been decentralized because of what's happening in Syria. So it has been decentralized to different places. And it happened that he moved with the group of scientists to Rabat, working on crop improvement, plant protection, etc. Now, of course, it is very important to show you the expected changes in precipitation and temperature, because that's very much related to insect development, even insect expression of resistant genes, because they are affected by temperature and humidity, etc. So we are expected, this Ioana region expected to have a reduction of 20 to 30% in precipitation, and that's a lot. That's a completely new environment that we'll have. The temperature, of course, in this region will also be elevated by between 2 to 4 degrees C, and that's a lot in terms of expression of resistance, durability of varieties, etc., etc. Now, I will mention there are several insects that are, I would say, important on cereals and legumes and cactus, etc. So I will describe a little bit to give you a flavor of the insect we are working with, so that, and then I'll be sharing with you what we have done when it comes to host plant resistance. So this is quickly the some pests. I would say the #1 pest of wheat in West Asia and Central Asia. It affects about 15 million hectares of wheat in this region. It affects both the vegetative stage and then also the reproductive stage. Of course, you get several seed if the infestation is high. Most of the damage is done to the grain, because the insect, as it feeds on the spike, it injects a protein in the protease that decomposes grain gluten. And then, of course, you will not be able to make good bread. This is the kind of bread you make from infected kernels. It is so, the enzyme is so potent that if you have only 2 to 3% of the grain lot is infested, the entire lot is ruined. The other insect I will be talking about is Hessian Fly. This is the #1 pest, I would say, in North Africa, north of Kazakhstan. Of course, I'm talking about our region, but it's also a problem in Spain, Portugal, in North America, mainly US. But I'm talking about, concentrating my talk about North Africa, West Asia, and Central Asia. So in Morocco, for instance, it is the #1 pest of wheat. I would say roughly about 30% yield losses caused by Hessian fly. You see here the susceptible material. If you plant late, we harvest nothing. Now, there is another insect, cousin, I would say, of Hessian fly. It's called barley stem garmis, same genus, by the way, Matula. It goes to barley, and so devastating on barley. Because of climate change, it has moved also to the Middle East. It used to be only in North Africa. Look, this is a barley field, completely damaged by the barley stem. And this is wheat, no damage at all, because it does not go to wheat, at least in our region. Russian aphid, it's an insect in aphid, causes leaf rolling, trapping of spikes. It's a problem in highland, basically in several countries of our region. This is a wood stem sawfly. We have, of course, different species occur in different places, but in our region, like between North America and North Africa, West Asia, we have completely different species, but the damage are similar. The insect cuts at the base of the plant, and then it goes here over summer and winter, and then in spring it emerges again. What it does, as you know, it causes these stems to fall on the soil. Then they will not be picked by the combine harvester when there is harvest, and that causes quite a bit of damage. Now, this Syria beetle is a pest of Central Asia mainly, on wheat and barley. I'm telling you this just to appreciate how did we come to know about this, the importance of this insect, because through surveys, we have done a lot of surveys in the region, field surveys, really to decide which one and conduct yield losses, etc., to know which of the pests is a key pest, is cause economic damage, etc. This is Syria beetle. You see here the insect reduces the green part of the leaves, basically reduce photosynthesis ability of the plants, and thus reduce, of course, yield. On legumes, there are a few insects like the chickpea bollworm. It used to be a problem in sub-Saharan Africa, but now it is a problem also in our region because of climate change. Faba bean stem borer, problem in North Africa, West Asia as well. On faba bean, this is the adult, this is the larvae, and this is the leaf miner. On chickpea, mainly on spraying plant chickpea. We have estimated yield losses caused by leaf miner on spring planted chickpea, it's 40%. That's quite a bit. Now, recently, Morocco got this Cochineal on cactus, and since 2014 to now, basically, cactus is almost disappearing from the country. It spreads so rapidly, and it causes a lot of damage. And I'll tell you the story where we are with this insect later on. Now, I do consider, of course, host plant resistance as the integrated pest management foundation. There is host plant resistance for most of the insects. You see here, this is the leaf miner, susceptible resistant. This is Hessian Fly, resistant, susceptible. This is Russian aphid, susceptible, resistant. This is cactus, Cochineal, susceptible, resistant, etc. Now, Painter in the early 50s defined really this. He was the one who started work on host plant resistance to insects, and in fact, he graduated from the same university I graduated from, which is Kansas State University. He was working there at Kansas State University, and then he wrote a famous book in 1951 on host plant resistance to insect, where he defined host plant resistance, where he defined the categories of resistance, etc. Now, this is a slide showing basically what we have in terms of genetic resources at ICARDA Gene Bank. That's very, very important. Without conserving these natural resources in the different gene banks, we are at risk. There are, of course, drought occurring so frequently now, flooding, fire, etc., etc. So this is why it's very important to collect and conserve these genetic resources in gene banks. So ICARDA has a wide, it has 155,000 taxations collected so far. And then, of course, different species. They even have rhizobium strains at this collection. So I'll tell you the importance, of course, of this collection in a minute, because here, the gene bank, this treasure, where we find resistance to the different insects. In relation to the gene banks, ICARDA and its partners developed what they call focused identification of germplasm strategy. Imagine 155,000 accession, how much time it would take a pathologist or an entomologist or virologist to screen, or a physiologist screen for the different traits. When I was at ICARDA, I was doing probably maximum 1,000 lines a year. And then you need to see how many years you need to finish that collection. This is why the ICARDA and its partners developed this strategy based basically on the environment, pest distribution in relation to the environment requirement for the development of the pest. And then there is a logarithm that is developed, of course, and then you get a subset from the gene bank where it is likely to find a resistance to your trait, pest or disease or whatever. The next slide, this slide explains a little bit in a simple way this fixed approach. The approach is based on the Darwin basis of selection or co-evolution. So there is selection pressure, meaning that the insect or the disease or whatever, the insect has been co-evolving with the host plant. There is co-evolution. There is adaptation, of course. This is the principle of Darwinian, principle. So the susceptible one, the weak, will disappear, and then, of course, so basically, use these principles. And for pests, you just select material from environment that favor the development of pest population, meaning that this is where there has been co-evolution, where there has been adaptation, where there has been selection pressure, and this is how you base your algorithm of this fixed approach. Now, there are ways of screening, and I need to tell you this because it's time. So that you appreciate how much time it takes really to find resistance to an insect. This is screening for Ashmatiac. For instance, we screen in the field, hill plots, or artificially infest because we need to make sure that we have infestation, and anything that's selected from the field, it goes to the greenhouse. This is unreplicated trial, and this is replicated trial. So that we make sure whatever we give to the breeder at the end of the day, we say, well, this is a source of resistance that is good. It is good. Confirmation from both field and greenhouse. So it's very simple. We just use a scale from 1 to 3 for leaf rolling, because there is a leaf rolling. This is a scale here. You see leaf rolling, chlorosis, necrosis, etc. And then there is a 1 to 6 scale for leaf chlorosis. And then we very simple to use. This is how we do evaluation for resistance to this pest. Now, we'll be seeing this slide as we go along with the different insect, because look at only at Rashmatiaphid here, the blue ones. So we have identified resistant triticum species. These are wild species, wild Aegilops. Also in synthetic, for those who do not know what synthetic wheat is, synthetic is that cross between durum wheat and Aegilops. Then what you derive, that would be a synthetic wheat. So there is quite a bit of resistance in the synthetic as well, and also find through figs the resistance in bred wheat and durum wheat. Now, see, I like to give messages here. I mean, I could stop in the slide before and say bye-bye, thank you so much. But to me, that's not the full story. Full story when you see your germplasm being used, that you have contributed to its development. And working with pathologists, we have developed material carrying resistance to Rashmatiaphid and resistance to yellow rust. Working with, of course, pathologists and breeder also, we have developed barley carrying diseases to measure barley diseases and Rashmatiaphid. And you see here, must stop and say this is our late Dr. Ajaram. He's, of course, the 2014 World Food Prize laureate, and he died because of COVID a few months ago. He was a fantastic breeder. His wheat varieties are grown in almost everywhere, every country in the world. And he's a famous person that we all miss very much. So, by the way, this germplasm, wheat or barley, has been shared with our collaborators worldwide. And that's you really feel happy to see your material that you contribute to its development is being used. Either to release or in crossing blocks at the national level or advanced research institutes, whatever. That's great feeling. Now, this is how we screen for Hessian fly. Very simple. We delay planting date in the field. This is Jamaat Haim. And then we excellent discrimination between resistant and susceptible. And then anything that, of course, we select in the field, we bring it to the greenhouse for confirmation. Very established system. We, by the end of the year, we have confirmed germplasm with resistance to Hessian fly. again, the evaluation, you have a very established system. The susceptible plants are dark green in color. And of course, they have larvae. The resistant ones are light green in color. And of course, they may contain dead larvae because on antibiosis, I'll talk about that later on. This is a dead larva here, and you see the difference. This is the live larva here. So this is mainly for Hessian fly. There is, of course, the larvae die when they start feeding on resistant plants, and then I can explain that later on. Now, this is the same slide you saw earlier, but just looking at Hessian fly now. Hessian fly is this color here. And then, of course, you have found resistance in Aegilops, also in synthetic wheat. And I'm talking about our region because there has been a large source of resistance in wheat identified to the Hessian fly in Spain or US, etc. Now, this is a very, I would say, a very successful story where a lot of partners, ICARDA, in Morocco, Kansas State, Summit, and so joint effort has yielded really the development of varieties that are now in farmer fields. And by the way, this is Morocco, by the way, was the second country to release resistant varieties after US. And that was a great achievement. By the way, the story won several national and international, of course, award and a lot of publication, of course. But it's great feeling to see your product that you contribute to its development in a farmer field. That's what a scientist, yeah, it's good to publish. That's great. For every input publication, they're all around for each part here. But it's also nice to see your product at farmer, grown by farmer. Now, this is one slide I will not show for all the evidence, just to show the importance of monitoring biotypes development. These are differentials. These are wheat lines with different genes for resistance. And then, of course, these are Hessian fly biotypes, Moroccan, Syrian, Kazakhstan, etc. And then you see the resistance to the different biotypes. That's very important information to have for the breeder. For instance, the Syrian Hessian fly is the most virulent fly worldwide, by the way. Only 2 genes are effective so far, only 2 genes. And so this kind of information is very good to give to the breeder for gene deployment strategies and also looking at evolution of virulence in the insect populations. I move to some pests now. This is how we screen for some pests. We developed, this is the screening technique that we developed at ICARDA. Big cages, 6 by 9 by 3 in height, where you can grow 200 lines. Here plot, we infest at 2-3 leaf stage, 2 adult per hill. That's a large insect. By the way, the threshold, economic threshold, only 2 adult per meter square. We put 2 per hill just to make sure you put live pressure on the insect. This is how we could discriminate between resistant, susceptible, resistant. And this is again susceptible, resistant, susceptible, resistant, susceptible, resistant. And then we have developed a scale from 1 to 6, very simple, 1 no damage, and then of course 6 more than 75% damage with severe stenting, like this one here. That's very simple. Now, again, the same slide, look at the some pest, this color here. So we have identified the resistance through fixed approach in synthetic, in bred wheat, and also in durum wheat. And of course, the breeders, I don't stop here. I give the breeders to make crosses. By the way, let me say this sentence here. When I joined ICARDA, when I came, the breeder said, Mustapha, you need to start screening for Russian aphid. I said, yes, with one condition. He said, what? I said, if I find, my lab, not me, if my lab finds resistance to Russian aphid, you need to use it in your breeding program, in your crosses. Otherwise, don't waste my time. And that was really the approach I use with breeders. I'm still doing it until now. And that's it helps. So this source of resistance, you will see they have been made, they have been used to make, to develop these mapping populations. We developed 2 mapping populations. Again, this is the role of a breeder. A mapping population is a crosses between susceptible resistant to advanced mental. They are fixed for resistance. And this is, you have different, not only for resistance, susceptibility, resistance, etc. That's a mapping populations. Phenotypic data, this is my lab did 3 years. Evaluate this mapping population for resistance to the samples. And then, of course, with the molecular biologist, he used the information, and then, of course, using different markers, etc. And then we were able to identify, and for the first time, a marker for resistance to samples. And this, by the way, has been validated on large number of wheat varieties and lines. And now it is, of course, made available to breeders for use in their breeding program. Because I'll explain to you, when you have an insect with 1 generation per year, you cannot read it in the greenhouse. It's very difficult. In my opinion, it is a must to develop markers. Otherwise, because working on, doing phenotyping in cages, in the field, it's quite, mainly when you have only 1 generation per year, it takes so long. It's very difficult, in my opinion. Now, this is another insect where we have only 1 generation, so 100% field screening. This is wheat-stem sawfly. Yes, we have found resistance based usually on the solid stemness of the stem, basically, of the wheat. And yeah, so we have few sources of resistance identified in bread wheat and durum wheat. However, you see here, these are the resistance for sawfly in bread wheat, in durum wheat. Not much progress, no crosses basically were made because it's low priority, seriously, in terms of damage importance. So, of course, the breeders should understand that. They have so many characters to worry about. So unless the pest is, so damaging, of course, they will do big crosses. Otherwise, but anyhow, we have source of resistance in case there will be need in the future. I move to Syrian leaf beetle. This is an interesting story. I was doing survey with a colleague pathologist from Icarda. We were in Kyrgyzstan. We came to a station, and then this station here, the research station, and then there was a huge infestation by the Syrian leaf beetle. Huge variability in a collection of wheat, 3,500 wheat lines. So I said, my colleague Amor said, Amor, you continue with the survey, I will have to stay here and evaluate this nursery. So I stayed, evaluated. We selected at that time with this woman here. She's a scientist from the station. We selected 266, but later on, because field evaluation, there is no confirmation. We've been testing until finally we took this 12 and did what you call category of resistance study under artificial infestation, and then we confirmed the resistance of 5 lines. Now this is a scale of 1 to 6 that you use, very simple, no damage. Of course, 5 would be 75% or less, etc., etc. So it's very simple system that we developed, and we are using it until now. Now, this is what happened with the categories of resistance that Painter called in 1951, basically categories of resistance, and we are still calling them categories of resistance. But he was confusing. He was saying mechanism, but now, he was calling them mechanism of resistance. But later on, we decided to call them categories of resistance. And then there are, of course, mechanisms of resistance where you go into biochemistry, phytochemistry, etc. Now, very quickly, I know probably not all of you are familiar with this type of studies. So these are the different lines. Mean of position, this is the number of eggs laid. This is a special test that we do, what you call non-preference or antagonosis test. And you see here, this susceptible chet has almost the highest number of eggs laid compared to this two, compared to this. And look at this, they're similar to the susceptible one. So these are susceptible. Look at the, for instance, survival rate on the different genotypes. High percent survival on this susceptible one, less on this, high on this. This is how we do. We look at we do categories of resistance to really confirm the resistance. Now, of course, we also use the feed and damage. And this is, again, this is susceptible check. This is what we call overwintered adult. This adult that stayed from the last year and new generation adults. But anyhow, these are adult, basically feeding studies. You see more damage by adults on susceptible check than on this here. So these are, of course, resistant. Now, I move now to screening for chickpea leaf miner. Again, it's a field screening because it's difficult to review insect in the greenhouse, even though it has 2 to 3 generations per year. We delay planting date in the field, and it's very simple. You can see nice difference between susceptible and resistant. Again, 1 to 9 scale, damage scale, based on the mining. This is what we call mining here by the larvae. And then this is how we categorize resistant or susceptible. This is again back to the same graph. This is chickpea leaf miner here. At that time, I will show you, we have identified later on more. But at least these two, they have been made. They have been used in crosses with by our breeder, Chickpea at the time. And then yes, he was able to. When I say crosses, they believe me, they were made. First one, they were made in 1999. And then, of course, it took all these years. This year, he was able to release some lines with resistance to chickpea leaf miner, and then with some relatively acceptable agronomic characters. And then, of course, this material has been shared internationally, of course, with our collaborators. Now, again, this is a slide, a very important slide to show the importance of collaborating with different teams. Look at this is 1992 genotype selected by FIX. This is our, the specialist from the genetic resources through his algorithm. He gave us this, I would say he selected the subset to test for leaf miner. My lab, over 3 years, did the phenotyping in the field. Now, of course, molecular biologists use the information, did his molecular, use different markers, and then he was able to identify a marker related to resistance to leaf miner. Again, this is a close collaboration. You cannot, one person cannot do it. So you have to have your geneticist, breeder, your entomologist, your molecular biologist, and then later on, of course, you could relate this gene or this marker related to this activity here, the gene activities, this glutathione gene, and it was confirmed on another leaf miner on vegetables, legumes and sativa, so basically same activity related, linked to this marker. So it is great. So this is the first marker we identified associated with leaf miner resistant chickpea, and that's again because of the difficulties in screening. It's very good to have a marker associated with resistance to pest. Now, this is another pest, another big one. I would say amphababine. It's called stem borer. We did lots of screening in the field, and different germplasm. Then, of course, the evaluation was very simple. It was based on percent infested plant. And then later on, we did screening under cages as well, artificial infestation, just to confirm. Because in my opinion, anything in the field, you cannot say 100% resistant unless you do it under artificial infestation. And that's what we did. So this graph shows you really the evolution of the material we've been testing over in the field. And then in 2017, we tested these lines under cages in 2018 and 19, and we confirmed the resistance of 2 lines. This is the first resistance to stem borer, fiber stem borer ever reported, and we are very happy with this achievement. Now, quickly, this is just to show you that we have identified, of course, I was showing you the other graph, but we have identified new source of resistance to the different pests whether stem borer or Hessian fly, leaf miner. This is nice. Leaf miner and ascochyta blight, leaf miner and wilt disease, and that's really good. But there are insects where we did not find resistance at all. So another message here, not everything can be solved with host plant resistance. So far, for chickpea pod borer, nothing really of interest to the breeder, or even in the barley stem garbage, no resistance so far. So I'll move to the last insect, which is the screening for resistance to the cochineal. As I told you, in 2014, this insect got into Morocco and caused tremendous damage. So unbelievable how it's like fire. I've never seen such devastating pest. So what we did, of course, there is a whole integrated pest management approach. Since the talk is about host plant resistance, this is why I'm concentrating only on host plant resistance here in this talk. So we screened an ENRAC collection in the field, and then also in the lab and also in the screening cage. The reason we did this in three is just really to confirm. Field, it was in a hotspot area in CW. It was naturally infested, but also we placed one infested cladode on each pest, basically cactus, on each ecotype. And also in the lab, we did the same thing. So with these pieces here of each cladode, of course, it was a replicated test, etc. We did the same thing, 3 replication under screen cages. This was in the field, but again, artificially infested. We were just putting again infested cladode on each pot, and that was really worked very nicely. And this is the grading scale we used: 0, no colonies attached at all, or sometimes even if they attached, they die, and etc. So this is the whole scale is based on how many. When I say colonies, I'm talking about this white stuff here. These are females that have been attached to this cladode. Now, in this study, we went and did what you call categories of resistance. This is what you call antibiosis test. Antibiosis test, looking at the negative, what definition of antibiosis, of course, this is the negative effect on the development of the insect. Mortality, slow development, etc. This would be antibiosis. So we looked at the nymphs, whether they are dead or alive on different test ecotypes. So look at the control. Of course, there was very little natural mortality compared to the other varieties or ecotypes that we identify. So very clear that there is antibiosis effect on the cochineal, confirming the resistance to reaction of the material germplasm to the cochineal. And this is a test, what you call anti-exosis or non-preference test. This is, again, what we are looking at the number. So when you place this is usually the way it is run, you plant your cladode in pots, and then you place them in the middle, you place your nymphs in a pot or piece of paper, and then let them go wherever they want. We just look at 72 hours after infestation. Of course, you do several replication, etc. And then, of course, you see, like for instance, look at the control. Significantly higher number of nymphs went to the susceptible control compared to, of course, the other ecotypes that are resistant. So these two tests show that mainly, of course, antibiosis, which is a negative mortality of the nymphs, that's really the most important mechanism of resistance or category of resistance or mechanism of resistance used by the cactus toward the nymphs development, and to a lesser extent, the non-preference, the way we see it here. So, of course, there are the resistant cactus ecotypes are less preferred, of course, than the susceptible ones. By the way, We are going to go to another step further. Now that I'm at UN6P, in collaboration with our colleague from INRA, UN6P, we will go into profiling these different susceptible and resistant ones to look at the, understand why, what are the chemicals that are responsible for, say, for the mortality of the nymphs, etc. We will go into an area of looking at the volatiles later on to see when it comes to non-preference, whether there are some volatiles that are also used to make this cultivar or this variety or this ecotypes less preferred than the other one. But and then also, since I'm here, this part of this collaboration with INRA, UN6P, we'll start a genotype and we'll genotype the whole cactus collection. At INRA, UN6P also has a small collection, and we'll do genotyping, and then we'll see whether we are talking about different genes for resistance. We look at the genetics basically of this resistance. Now, again, we have identified for the first time, of course, in Morocco, first source of resistance, even at international, not many source of resistance really identified. I'm talking about cultivated, for the fruit production, cactus producing fruit, not really fruit producing cactus. We don't have much resistance identified. And so we're happy to find resistance to in the INRA collection. Excellent, very fruitful collaboration between INRA and the CARDA. 8 ecotypes have been found resistant. They have been registered in the Moroccan resistant catalog and are being multiplied in several platforms for distribution to farmers. By the way, this team just won the Prix Hessian the Grand Prix Hessian declared just about 10 days, 2 weeks ago, one of the categories, I believe in innovation, whatever. So I think that was great to have this. But again, it's good to win, publish several papers, but good to win the award. This is again in collaboration between INRA and CARDA. But the most important is to see cactus in farmer fields, hopefully in the near future, to replant what has been devastated by the Cochineal. I'm almost to the end, Mr. Chairman. I'd like to thank, several of our partners, the CARDA, CIMIT, national partners in North Africa, West Asia, and Central Asia. For instance, there are some key partners like in Morocco, for instance, etc. And then we have several advanced research institutions from Canada, US, Australia that have been part of this effort on host plant resistance to insects. Of course, without funding from different donors, we would not have been able to accomplish what we have really accomplished in terms of germplasm development, in terms of characterizing the different pests, etc., and doing the screening, etc. So several donors, FID UK, USA, the OCP Foundation as well, EU, IFAD, Germplasm, Australia, etc. And now I'm almost done. We've been talking a lot about food security. That's fine. But I think we have been, our colleague breeder Muez and Michel Ghanem have been talking lately, this group, about nutritional security as well. That's very important. Produce orphan crops, some of the crops that have really good nutritional qualities. But very importantly as well is to have the safety should be considered. Because when we develop host plant resistance, develop germplasm, doing breeding, of course, we are not spraying pesticides. When we develop, use biological control, we are not spraying pesticide. We are using plant extract or biopesticide in general. We are not using pesticide. And these are, I'm hinting to some of the areas I'll be doing with my colleagues here at UN6P and partners elsewhere. So looking at the safety, develop safe food, but also, of course, with the colleagues here and elsewhere, nutritious food as well. And I will, let me just remind all of us that we should all keep in mind the smallholder farmer. And we're in CDFNI with my colleague, Dr. Sbahi from INRA. We're in CDFNI in the South Morocco. We did survey. This was a year and a half maybe ago. And then this gentleman saw us and he said, "What are you doing here?" So we explained to him. And then when he finished, he said, "Okay, bye-bye." He said, "No, you need to come with me, have food." He insisted. It was not possible to escape. So he came to his place, excellent tagine. I don't recall whether it was from goat or not, but it was really good. And the homemade bread, and also the dessert. There was, of course, cactus fruit. But imagine with the cochineal, if you do nothing, there will be no fruit, no cactus as dessert at the for lunches or dinner, whatever, for a lot of people. But the other time, we need always, as scientists, to keep small farmers. That should be one of our targets, is to really improve the livelihood of the small farmers. There are a lot of small farmers out there in the whole Africa, since we started in Africa, that need help, that need our contribution to really improve their livelihood. And with this, I would like to thank you. Thank you very much. I'll be happy to entertain questions if you have.
Thank you very much, Professor El Bouhsssini, for this very insightful webinar tackling a very important subject of the host plant resistance to insect pests in North Africa, the Middle East, and Central Asia. And this webinar has been viewed by about 50 some viewers from different countries, from Ghana, Senegal, Nigeria, Kenya, Ethiopia, aside from viewers from Morocco, of course. And I'll take this opportunity to salute our friends in Nigeria, because they were so numerous, identifying themselves as such, sending their greetings from Nigeria, and thanking Dr. El Bouhsssini for his interesting webinar. And I hope they have learned something through this experience from a bright entomologist in, I guess, in the world, because he has been in Kansas State University, America, North Africa, the Middle East, Central Asia. And why not, as a plant protection specialist, let's try to initiate some movement among the plant protection specialists in Africa, in African countries. And we build, I believe there is an association. Let's investigate that, and let's see, and let's join it, and let's fortify it, and make it very Africa-oriented and resourceful. That's my wish. Now we have some questions and comments. As you might expect, two of these questions and comments are from our friend Muiz, whom I thank very well for being an assiduous viewer, participant of this webinar. Thank you very much, Professor Muiz. And he says, chickpea pod borer, Helicoverpa armigera, remains as one of the most challenging problems in the dry areas. No resistance sources were identified so far. What would be the best option to control through IPM approach?
Thank you, Muiz, for this very interesting questions. As I said in my presentation, now I believe, so not for every single insect there will be, or there is resistance. We have tried even figs approach. We have tried different corrections. Well, if you talk to ECRISAT people, they may say that they have some resistance in the wild relatives, some tolerance. But my opinion, we should probably focus on management approaches. And there are, of course, several. There are biopesticide based on virus formulation, on plant extract, etc. So I don't, yeah, that's, so we don't have to rely only on host plant resistance to insect to solve problems. Just see, pod borer on chickpea would be like probably almost like Rovanki on Fababi. You have some partial resistance. Yes, there is some partial resistance at pod borer. This is what our colleagues from ICARDA worked at my very good friend of mine, Dr. Harris, did at ICRISAT. But again, I don't think it's an acceptable level of resistance that one could use in breeding programs. So this is why it has to be, we have to look at other components as well, biopesticide. Biopesticide, there are different, as I said, viruses, plant extracts, etc., etc. Even cultural practice could do intercropping with some coriander to encourage beneficial insects, etc., etc. So that's what it is. But I would say host plant resistance. But remember the way I defined host plant resistance should be the foundation only. The foundation, it may not be enough. You have to complete with walls and roof, etc. So it's nice. Yeah, probably did not have time to talk about that. Two things. When you have, even if you have 100% resistance working, you should worry about biotypes, because variant biotypes would develop. So this is why it's good to have other management approaches used so that you reduce the pest population. So the more you reduce the population, the less chance you have for, of course, mutations are occurring spontaneously, but at least you have a reduced population by host plant resistance, by other management options, so that you decrease the chance for those different alleles to start mating and then increase in population and become a biotype. So this is why, even if you have resistance working, you have to go with a management, an IPM approach.
Okay, thank you, Professor Bouhsssini. Maybe I don't know whether these statements and conclusions are valid also for the chickpea leaf miner. Here is our friend Moiz again with another question about the chickpea leaf miner. And he says it is more a big problem for spring chickpea. Many resistance sources were identified and leaf miner lines were developed. We should explore more the mechanisms of resistance involved.
Yes.
This is his recommendation.
That's correct. This is what we have been doing all the time, looking for, by the way, for chickpea leaf miner, there is resistance is very much related to oxalic acid concentration. So you could do screening basically at your lab, and the higher the oxalic acid concentration in your lines, the better resistance you have in your material. Now, leaf miner is not like pod borer. There is a good, very good level, I would say, in 1 to 9 scale. You have 2, 3. That's good. That's, I would say, acceptable level of resistance that a breeder could use. But again, use with other, go with early planting date. You don't have to worry much about leaf miner. If you go to early planting, like winter, fall planting, whatever, early planting of chickpea, you reduce by half infestation by the leaf miner. But of course, if you delay to spring, then you get more. And if you delay, then use other. There are some botanical pesticides, etc., that you use in addition to resistance.
Okay, the next comment is from Dr. Carlos Iglesias, North Carolina State University. Thank you, Carlos, for being with us for all the webinars. Thank you. Please convey our greetings to all the faculty of North Carolina State University. Carlos says, how would pests evolve in the coming decades in front of climate change / instability in North Africa?
Yeah, that's a very good questions. I did a whole seminar on this sometime, but I might give it again some other time. Yes, if you recall what he showed, there will be in the next 100 years or 8 years from now, I would say, there will be about 20 to 30% reduction in rainfall, and also increase in temperature between 2 to 4 degrees C. 2 to 4 degrees C, that's a lot for, in terms of temperature. So what it means for insects, we have seen examples. I don't want to predict. The chickpea pod borer did not used to be a minor pest in North Africa. It is not anymore because of climate change. We have now Hessian fly, for instance, moving north from North Africa, now Spain, Portugal. It has been found in France, see, because of worm. The example of the barley stem gall midge on barley in the Middle East. I've spent most of my, career in Syria, never seen barley midge. In 2000, believe 9, 10, 11, 12, major problem with the barley midge. So what we did when we examined the winter, it was warm. There was basically no freezing temperature for 4 years, and that really favored the development of the pest. There was no need to go for overwintering, diaposing, etc. It was multiplying, and then it has become now a major pest. Yeah, climate change, I would say, is with us. So what's going to happen in general, in my opinion? Some would leave some places and then settle in another based on because different insects have different range of environment where they really prosper. So so you will see some in one region, some minor ones in another region become more important, etc. It has started. You have started seeing that, in fact, for insects.
Okay. Our distinguished colleague, Professor Michel Ghanem, congratulates you on your talk. He says, great talk, Mustapha. And his question is, how do you see coriander integration in Moroccan cropping system?
Very good question, Michel. You see, of course, I favor integrated pest management, even though I spent most of my career on host plant resistance. Now, when it comes to intercropping, coriander could be just an example, Michel. In my opinion, we should assess the medicinal plants in general, and then propose flower and medicinal plant into different cropping systems. Coriander may not, I don't know whether it would be suitable to incorporate into the cropping system, because it is very, very important crop for natural enemies, for beneficial insects, for egg parasitoids. It has big flowers. The flower is full of nectar with the 3 major sugars. The natural enemies, parasitoid, they love them. Without this kind of flowers into the cropping systems, we will not be able to have beneficial insect into our cropping system. But again, I cannot say coriander or any other species. Every country needs to assess the different flower and species they have, the medicinal plants they have, and then do propose intercropping. Because intercropping has two implications. One, an income generating technology, because of course it's a cash crop, additional income for the farmer, but also good the flower are very good for the beneficial insect in general. But again, I cannot recommend this species or that species because it based on what's really good in that region, based on the environment, etc., etc. But the most important thing are flower and medicinal and adapted flower and medicinal plant or few medicinal plant into cropping systems.
Okay, thank you, Professor Bouhsssini. And the next comment is from Mr. Patrick Kimararungu. I suppose he's from Kenya, if I'm not mistaken. And he says he has a question concerning the control strategies to pests pastoral, which move from one place to another, for example, recently in Kenya, Uganda, and Tanzania. What countries can do?
Well, they can do a lot, of course. I mean, one of the things countries should do is to establish some early warning systems, say, across regions in Africa or whatever, or wherever. Because to me, without that, you will be faced with a, here is a pest, and then you start wondering what to do. So with the climatic data, with the biological data of the pest, etc., then you can predict where the pest would move, which direction, like diseases. They have, it's a very good system they have for UG99 with the rust, with the FAO, ICARDA, CIMIT, and several other countries that are part of this. So on insect, I must say, we're not that sophisticated. I would say we're not that prepared for insect. And seriously, just recently, I've been discussing with some colleagues from Amstead, etc. We need to establish some work on establishing monitoring prediction systems, for some key pests in Africa.
Okay, thank you very much, Professor Bouhsssini. Our next comment is from Mohammed Aziz Al Humaizi, and I suppose he knows you because he has been sending greetings. So he says, nice presentation. Presentation is important, but it includes the trees, then the herbaceous plants. It includes the trees, then the herbaceous plants, annual. And how predict the disease development by climatic and soil data to better fight against the disease in IPM? I guess
so maybe
question. Maybe you have gotten the sense of it.
Yeah, maybe he was saying the opposite. I concentrated on annual plants than on trees.
Yes,
that's what they did. Yes, you're right. I did not want to talk about, I have some experience on date palm, but in my opinion, it's the same system. You are talking about how to use the climatic data, soil data to predict. And this is what I was referring to in the previous question. Yes, we need to use all the climatic data, whatever data is available, biological data of the pest, really to predict where diseases and pests will be moving, so that we are well prepared when a pest comes, etc. Because I think what we are doing now, a pest comes and then we start, what to do. So I think we need to go a step further and start developing, I would say, work with our modelers using survey data, because we have done a lot of surveys. Every country has done a lot of surveys, and we could use those survey data really to plug them with climatic data, survey data, etc., to predict new move of pests, diseases in different countries, so that we are well prepared.
Okay, thank you. Here is another, maybe the last comment from Jean Damascene Touyezer. He says, thank you for your excellent presentation, doctor. And he has a quick question, and it says, how do you relate soil fertility and the ability of crops to resist to pests? Thank you.
This is very good question as well. Now, let me tell you the following. I have seen, been with colleagues from INRA working on demonstration plots of resistant variety to Hessian fly. And seriously, I could not, there were cases where you could not see the difference between susceptible variety and resistant variety to Hessian fly. Why? Because the two were full of weeds, no proper filterization, etc., etc. Anyhow, in my opinion, of course, when you have good soil fertility, I would say adequate source of fertility, then what you do, you have, even if you don't have resistance, your tiller, the main tiller might die, but with proper soil fertility, you trigger tillering, and this tillering would grow faster, and then they would escape if there is another generation, whatever, of the pest. So definitely, of course, there is no relation with soil fertility, I mean, per se, and resistance, but I would say tolerance, yes. So the more you have proper management of your crop, soil fertility, weed control, whatever, the more that trait, the resistance will be expressed, and you will see the difference between really susceptible and resistant ones. But again, even if you have only susceptible variety, use proper, use other management. So fertility is one of them, so that you encourage tillering, growth, etc.
Okay, thank you, Professor Bouhsssini. I cannot help but to post this last question from our friend Dr. Ahmed Tahbani, one of the ESAFE's professors in entrepreneurship and business. And it's good to have you, Dr. Tahbani, among us. And he says, thanks, Dr. Mustapha, for this presentation, referring to the cactus mania in the mid-2010s and the burst of the price bubble added to the cochineal issue. Is there any revival perspective for cactus? And I suppose that he refers to the Morocco, but especially to the Rahanna region.
Oh yes, definitely. Now with there's not only resistance that is available now. As I said, the resistant cultivars are being multiplied in several, more than I would say 15, maybe 20 platforms in the country. There is plenty of material that will be available to farmers, I believe, next fall. And this is one. Second, we did not, the team, INRA ICARDA, did not work only on resistance cultivars. We also worked on biopesticides, on biological control, etc. So yes, there is hope. I would say we have several products good product biopesticide that could be used to manage the cactus. And this is one. Second, also, as we speak, there are several students still working on the cactus cochineal. And also here at UM6P, there are, when I came, I found several scientists working in the valorization lab, working on plant extract, other product for the control of the cochineal. And there are some promising products that are very, very good. But anyhow, I think I'm very optimistic. Definitely, we will have cactus back to normal in the whole Morocco. It will take some years. It may take between, I would say 5 years or so to have the whole, cactus back, I would say full speed in Morocco.
Yeah, I'm very happy to hear that. And I would be anxious to form squads from UM6P, students and staff, to go out and help in the planting of cactus, those cactus varieties that are resistant to cochineals. And I think this is a golden opportunity for us to link with the local farmers and share with them their germplasm. I don't know what the policy is now, and I guess our duty now is to to transfer the knowledge and the genetic material to the farmers and see how it goes. And this is an opportunity that we should not miss by bringing our contribution to the farmers who are stranded, who have lost all their cactus plantations. And we know that cactus, they offer not only fruits as a dessert, but they offer basic meals in the morning and at lunch and before you go to bed. That's a major food in areas where you cannot have any other crop. And I think this is our mission, to do something about the Rahamna region and the other neighboring regions first before we move somewhere else. And with that, I will thank you very much for this interesting seminar, webinar. And I will thank our viewers from different parts of the world who are assiduous to our ESAFE webinars. And your participation really is an excellent reward for us. We're happy to see you numerous, connecting in, exchanging with our scientists, and we hope for this academic year, we still have one webinar left on July 13th. And then maybe we will merge with a larger system of webinars at UM6P in order to allow you to a broader exposure from various disciplines being taught and various research topics being investigated at UM6P. I have no advertisement to make about our master's fertilizer science and technology program, because the deadline have already passed, and we are now we have obtained about some 200 candidates for 20 seats that we will screen from the best of the best. And I hope the very good luck to the best ones to be retained in our program next fall, inshallah. And that's it. With that, I will close this webinar, because our guest, our distinguished guest, has an obligation to join another meeting. Professor Mustapha EL BOUHSSINI, thank you very much, and we hope to see you some other time. And we are happy with your contribution.
Thank you so much for this opportunity, and we'll be happy to. It's my pleasure to share.
But do not forget the farmer field schools in Arhamna to plant some cactus.
Inshallah.
Okay.
Thank you.
Okay.
Thank you so much.
Thank you very much to all of you, and we hope to see you some other time. Bye-bye.
Bye.